The aim of this experiment is to see the impact of a test species of algae, that's Stichococcus, which is commonly found on building stones in the UK, on two test sandstones, that's St Bees and Dungannon, which are found especially in the northern parts of the UK. So how they respond to algal growth, and if the growth of the algae causes deterioration to the stone on which it grows. We'll be measuring the growth of the algae over 1 month in conditions of maximum yield, by which I mean climatic conditions which are favourable to algal growth. First of all, we have to measure the stone as a reference. So we measure hardness, the level of moisture in the stone, the colour of the surface and its weight prior to inoculation with the test algal species. The way we measure the moisture content of the stone is using a device called a protimeter. This measures resistivity as a proxy for the amount of moisture in the stone. Originally, this device is used to measure moisture in timber, and we've basically brought it in to use for this study. It's important to be thorough with the moisture measurements before, during and after the experiment. If we take 9 measurements across the surface, you can plot the distribution of moisture during and after the experiment. These can be inputted into a stats package, and we can end up with moisture distribution charts like this, indicate that the wettest part of the stone in the previous experiment was the central zone, with moisture decreasing towards the outer edges. Another measurement important in stone assessment is measuring stone hardness using a device called an equitip, which is traditionally used to measure the hardness of different levels of concrete. That the equitip works is that the prong has a small ball bearing in it which drops down to the surface. Across the surface we're going to take 16 measurements. This will form an average of the total hardness of the surface, and it will also give us precise measurements at different points on the surface, so we can correlate this with the amount of growth later on in the experiment. We can see if the algae causes deterioration as it grows. The final thing we need to measure is the colour of the stone, and we can use this as a proxy for the amount of algal growth. So the more green the surface, the more algal growth. We start with a blank measurement of the stone. The way the spectrophotometer works is that it shines a light onto the surface and it measures the irradiance you receive back. The surface colour at the moment, before the experiment starts, is very homogeneous, but we expect during the experiment that growth will be uneven, so we can compare this to the target initial measurement and see how it's changed. The next stage is inoculation of the stone blocks. The species we're using for this experiment is Stichococcus bacillaris, which is a common green algal species on UK stones. So this is a pure culture. For the purpose of the experiment, we've made a subculture and weakened the solution to replicate the kind of level of growth you would get in the real environment. I'm going to use a micropipette to pipette 3 millilitres of the weaker algal solution onto the surface. 3 millilitres is enough to cover the surface equally. Once we've inoculated the blocks with the algal solution, it's time to put them in trays. So important, it's necessary to separate the controls from the inoculated blocks and then put them in the growth cabinet under the right environmental conditions. Over the course of the month of growth, we'll be measuring how the stone changes, so weekly measurements and half-weekly measurements. And correlating these, we can get an idea of how the stone responds to the growth. So whether the stone is deteriorated by the growth or, in fact, if the algae is protecting the surface. Every week I've been measuring the stone bioreceptivity, so that's how the stone responds to algal growth on top of it. Over the month that the stones have been inoculated and in the growth cabinet, hardness hasn't changed very much as we would expect. Surface colour has obviously changed quite considerably, and it's a good proxy measure of how much colonisation there is on the surface. The outer edges are more colonised with little growth in the centre, and that's a common pattern on all of the blocks. Moisture measurements have indicated that the highest level of moisture across all of the blocks is in the central zone. And interestingly, this is the area of the blocks which has remained uncolonised. And that's primarily because this species of algae isn't tolerant to very high levels of moisture. These results demonstrate that this species of algae and similar species like Chlorella vulgaris, which we've looked at previously, favour specific conditions of moisture, humidity and temperature. And this is important because it can give us an idea of why certain species grow in certain areas of buildings, and consequently we can see if they cause any damage to those areas where they colonise.